Good evening, everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Hi, I'm Scott Sorrell. I'm going to be your host tonight here at Real Science Exchange. We're here to welcome Dr. Todd Callaway from the University of Georgia to further discuss probiotics and their impact on animal health and performance. Dr. Callaway joined us for the Real Science Lecture Series and presented a great webinar back on June 4th titled, Are Probiotics Just Magic Foo-Foo Dust? If you'd like to watch that webinar, go to balchem.com/real science and scroll down to the June 4th webinar, and you'll be able to click on it and watch it from there. Dr. Callaway, welcome. I believe this is your first time to the pub. And as we like to ask, what's in your glass tonight?
Oh, thank you very much. I appreciate it. See, today, literally, because I'm in the office, is water, because UGA doesn't allow us to drink on the job. We get in a lot of trouble for that. But normally, it is a bourbon like Bullet.
Bullet
Okay,
bourbon.
there we go.
All right. So, Dr. Callaway, I see that you've brought a guest with you tonight. Would you mind introducing him and tell us a little bit why you selected Dr. Steele to join us tonight?
Okay, yeah. So this is Dr. Michael Steele who's with us from University of Guelph, and I chose him because we wrote the paper that all this was based on together, and it all started from back. And correct me when I'm wrong, Mike, about being right before the pandemic. We started writing this review. We met at ADSA the year before, kind of hit it off. Mike came and said, "Let's do something together." Okay, so we started working on this, and then the pandemic hit, and it just everything, how everything was just nightmarish. Of oh, we've got to get this done. There's a million things on fire, and it took us forever to get it written. And Mike was so patient with me being behind on things, and he's always like, "Come on, you can get this done. Come on, idiot, you can finish this." And that just became how we did things. And we finally got it through, and I think it turned out to be far better paper and a good kind of overview of where we are in the industry of probiotics. And you know obviously, Mike, you can do a better job introducing yourself than I can. Clearly.
Yeah, thanks, Scott, for this invitation. But Todd, I really appreciate this. It's been fun working with Todd. He's such a humble guy and lots of fun to be around. So I've really enjoyed this experience of working with him on this review. And I think I learned a lot from Todd, and I continue to learn a lot about microbiology from Todd. I'm not personally a microbiologist. I'm more of a nutritional physiologist that dabbles in the fields of using probiotics and prebiotics in experiments. So it's always great to partner with a microbiologist doing this work.
Yeah,
Very
and that's been
well.
the secret to my career is I find people that are smarter and better than me, and I kind of help them, and I stand there and go, "Look what we did." So that's been my whole career. So Mike
Well,
is the next victim. I mean, partner in that.
Mike, what's in your glass tonight? I have to ask you
Yeah.
that.
So I'm calling in from the Netherlands right now. So it's the afternoon. So I thought,
Is
why
that
not? I
right?
have a Belgian beer here,
Yeah.
blonde.
Ooh,
It's 8% though. So I haven't started
Yeah.
drinking it yet. So just be aware that towards the end of this podcast, things might be a little bit more fluid,
A little
okay.
spicy?
a little spicy, but we'll see.
Yeah,
Is it one
good.
of the Trappist ones or
I
just a regular beer?
It's actually this new one from the Ardennes area of Belgium. So I started to drink Belgian beers since moving here, just on sabbatical, and yeah, wow, that's all I have to say.
Now they got some tasty beers over there. I like Duvel and Palm.
Yeah,
They're
me
two of
too.
my favorites when I go over there. So, all right, and I can't forget my co-host tonight. We're got Dr. Ryan Ordway joining us. Ryan, good to see you again.
Yeah.
Thanks for joining us. And got to ask, what's in your glass tonight?
Well, I am drinking of course, I'm still on the clock here, and it's not afternoon yet, but
It's
I,
always
I
happy hour at the Real Science
I'm
Exchange, Ryan.
I'm just I've got a glass of water, and I actually just got a fresh cup of coffee. So you can see that I've got the. I don't know if any listeners have young kids out there, but I even though my kids are not as young anymore, I'm still a big fan of Bluey. So I've got a Bluey mug of Nespresso coffee. So,
All right,
but
there you go.
I've gotten into rum in the last basically during COVID. So if I had my choice, I would be drinking a Diplomatico rum with a big ice cube. But
Well, I am having a local whiskey. My wife took me to a farmer's market this weekend in Narrowsburg, New York, and there was a local vendor there, and he had a single malt whiskey from Rock Valley Spirits. Had some last night, liked it. I still think I'm more of a bourbon drinker, but we'll see if, as I have this, maybe I'll begin to like whiskeys a little bit more. But one of the neat things, the other story I have to tell tonight. So I'm drinking this from my Glen Cairn glass, and I got this, and I've talked about this before in the podcast during a bourbon tasting at the Cavalier Hotel in Virginia Beach, which is an old historic hotel where that existed during Prohibition, and I did a bourbon tasting there. Well, my son took a job last week, and he's working for an ad agency, and the Cavalier Hotel, and specifically the distillery they have there, is his primary account. So I thought that was pretty cool. So I had to pull out the Glencairn today. So anyway, folks, looking forward to a great conversation about probiotics and foo-foo dust and the like, and dispelling those myths. So let's raise our glass to a great conversation and to some great people tonight. Cheers.
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 New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
So, Dr. Callaway, I'm going to jump right in. As I mentioned, that you participated in the Real Science lecture series. We had over 220 people register for that webinar, and we've had over 200 so far that's actually watched it. There's quite a bit of interest. People are looking for more information on how to improve the microbiota in ruminants. And relative to that, it's been my experience that there's a great deal of confusion and, dare I say, skepticism around DFMs or direct fed microbials. You've got probiotics and prebiotics and postbiotics and eubiotics and on and on and on. And so, as my first question, can you kind of help clear that up for us? What are the differences in some of these direct fed microbials, and are they all really relevant for feeding to ruminants?
Well, yeah, they are. It's the terminology has kept changing. For years, it was direct fed microbials, and Mike, correct me when I hit something wrong, but we called them DFMs just because it was easier. Because humans did probiotics, we did DFMs, and never the twain shall meet. But over time, as we've tried to figure out how you separate out these different modes of actions, we've had to develop terminologies that make more sense and are more related to their modes of action, or what we think generally their mode of action or composition is. So the umbrella term that kind of now replaces DFM is eubiotic. And what that means is it's anything that's going to support animal performance or health. So that can include our traditional DFMs, what are called probiotics, prebiotics, postbiotics, but also phytochemicals, organic acids, and essential oils. So it kind of covers this whole breadth of things. Then underneath that are probiotics. We've gone back to that old definition of probiotics, that is a living microorganism that is going to be beneficial to the health or performance of the host. So that means it's got to be a live bacterial, yeast, or fungal culture usually. So you can have your lactic acid bacteria, you can have aspergillus, any kind of the lactate utilizing bacteria. You can have just about anybody in that cluster as long as it's alive. And that also includes like the bacillus cultures, because those are spores, and they are, this is where your definition of what's alive versus not becomes real sketchy, because the bacillus are typically thought of as being spores, which is like a spaceship, a space capsule for the DNA, that when it gets to good conditions, it vegetates and starts to grow. So those are lumped in there because they're supposed to come back to life to be their mode of action. Then you have prebiotics, which are fermentable substrates that the host can't use, but the microbes can. All the oligosaccharides and all those fun things, fructooligosaccharides. And they play a role in ruminants, but it's really, the ruminant makes those really challenging and expensive to use. Because number one, you've got that large rumen volume that you've got to get your concentration to, but also because that rumen population is so biochemically adept at breaking things down, we can throw TNT into a ruminant and it will degrade, rocket fuel, you name it, the microbes will eventually eat it. And, you've got animals working at the Aberdeen Proving Grounds eating the grass instead of putting a private on a lawnmower, just turn goats loose. If a goat hits a mine and blows up, it was just a goat. We have barbecue now. Or if it finds unexploded ordnance, but they're able to cut the grass, and the microbes in their rumen can degrade like octanol, DMG, TNT, all these really nasty compounds that are leaching out from 100 years of explosive testing just fine. So, it's the great power of the ruminant, but the problem is those prebiotics, there's somebody in there that can eat a prebiotic. So unless we can kind of protect it to get to the hindgut and have some more action down there, they're a little bit iffy somewhat. But a lot of our postbiotics, which are yeast and fungal products or products of their fermentation, and they're non-living. So it can be the cell wall products, cell wall contents, all those things. A lot of those actually contain prebiotic or prebiotic-like compounds that work in the rumen and work in the hindgut. So the line that we like to draw of this is a postbiotic, this is a prebiotic, is a little sketchy there between postbiotic and prebiotic. I mean, what do you think, Mike?
Yeah, I would agree. I think you have some really good analogies, but we're very good at making these definitions change constantly, I would say. And I'm trying to get away from the direct fed microbial definition. I've managed to replace it with microbial-based solutions to cover all the bases. And I find that helps with the review process with certain papers that we're submitting, but also the symbiotics too. And I think that's a really interesting, at least when it comes to application, is the combination of predominantly pre and probiotics that are fed at the same time. I think that's one definition that we should include. But yeah, I think your definitions were totally correct. It's just you have to stay on top of some of these definitions, and they're constantly misused too. Um, are there applications too of all these? Yes, there is, but I think there's lots of opportunity to improve our definitions, but also the research that we do with them. And I think that's probably why they've received somewhat of a negative reputation sometimes, at least in the history. So lots of work to be done.
Well, that's where we came up with that title of foo-foo dust in the first place, because so many products out there are just, you give this to your cows, they're going to drive your tractor, they're going to shovel the pens for you, they'll date your daughter, they'll have clean teeth, their hair will look good. I mean, there's so much complete crap told to producers
Yeah.
and, people and other nutritionists that it's like, okay, if it can do all that, why aren't we president already?
Yeah, and it's easy to register some of these products without any research too. And I think that's over the last couple decades has been detrimental to the reputation of some of the more proven direct fed microbials or probiotic, prebiotic, postbiotic technologies that are out and available for the dairy industry.
There's some companies that they'll have 10 times the marketing budget that they do for R&D. And they wonder why nobody believes us. They take our free stuff, but they don't believe our product. I can't imagine why.
Yeah, I always tell farmers and nutritionists that you need at minimum, and this is like bare minimum, 3 publications in a peer-reviewed journal showing efficacy, and it has to be a really high-quality journal. Otherwise, I'd really be skeptical of what's on the market. But luckily, I think more and more companies are moving this direction, publishing in peer-reviewed journals and even publishing when it doesn't work, because guess what? Most of the time, it doesn't really elicit a response because there's not enough of a stress challenge. So I think that's really important to also show that under certain conditions or in certain environments, it works better. And
Yeah.
it's not going to be a silver bullet.
And that's been one of the big things that, Griswold's hammered for years about, what's more important than which one works, but why don't they work every time? Why don't they work in every cow? And, your point exactly, where they're stressed, either through production stress or environmental stress or a health challenge or whatever, what's going to affect those cattle and why doesn't it always work? And, you're down to looking at the bulk tank where you're trying to figure out an effect from 150 cows out of your 1,000 cow herd. It's hard to see a difference in your bulk tank level milk production.
That's one of the takeaways I had, Todd, when you were going through your presentation, is that you were talking about that maybe every dairy is going to require a custom blend or solution, which can be complicated, but maybe you could talk through that just a little bit.
Yeah, because you're going to have to understand what the challenges on each farm are, or what's the diet. Because, down here, what we're feeding is very different than what they're feeding on Mike's farm up in the Northeast. Well, way Northeast, but, you got different problems on each farm. Our animals are struggling right now. Yesterday we were at 95 degrees, but the humidity temperature was 110. So our cattle are struggling. It never gets below about a 90 feels like temperature here this time of year because the humidity is so thick. So our animals are under a completely different stress environment than cattle in Guelph or out in Montana or Wisconsin. So you're going to have to look at those, kind of regional level issues, but down to, where's your water coming from? Is your water cleaned? Is it not a very clean water supply? Are you pulling water out of a river that you have problems? So you've got some pathogens coming in. Do you have a lot of birds coming in bringing salmonella into your herd? Do you have, what kind of level of challenge do you have with those pathogens? What's your grind size? We made my grad student made that great discovery this year that grinding corn causes more acidosis than not grinding corn. What's your ration look like? So there's a lot of things that go into looking on a farm as to what's going to help you. If you're on a primarily forage diet, using a lactate utilizing bacteria is going to be pointless. You're not going to get enough lactate production for that to work. Mm-hmm. So, you want to look at what are my goals for this herd? There's a lot of herds that'll be fairly similar in regions, but you got to understand what are those drivers. What do you think, Mike?
Yeah, I think just adding and building upon what you're saying, Todd, is you need a somewhat of a challenge. I'll walk back to my first study I ever did looking at probiotics was 10 years ago. as an academic, and we did it on a wonderful farm with hardly any diarrhea or respiratory disease. The level of cleanliness was outstanding, and we found no differences whatsoever, even on the microbial communities. And we're wondering, wow, I guess this particular probiotic doesn't work. But no, it's because we didn't have a stress challenge. And then we immediately moved to a calf rearing facility where diarrhea is always well over 50% of them, if not closer to 100%. And that's where the same probiotic was actually showing differences with respect to incidence of diarrhea, resolution of diarrhea. So I think that it doesn't work in every single environment and every single farm, but specific ones have to be tailored to the challenge on that farm. And
And sometimes those challenges can appear out of nowhere too,
mhm,
like birds carrying HPAI onto farms.
mhm.
How do you,
Kind of pursuing this line of discussion, right? Our nutritionists out there today are very well schooled in customizing diets for nutrition. But where do they go to get the information they need to customize, their diets for, let's say, the microbiome?
Well, that's, number one, you use the magic word that everybody's got to take a drink to. When you say microbiome, everybody's got to kind of take a shot because, that's the word that everybody uses right now. And there's this idea that, oh, we can, and I hear this a lot of, oh, this will make your microbiome good. We did the first microbiome studies on cattle back in like 2005, first ones ever done in the world. I can't tell you right now what's a good microbiome and what's a bad one after doing this for almost 20 years now, because we don't know what we don't know. And there's so much going on. And I use an analogy a lot of during World War II. And I know, Mike, you're going to roll your eyes at this one because I've used this one with you before, but we'd send our bombers over from England. They'd go over, drop bombs, come back, and afterwards they'd do battle damage assessments on each bomber, where each plane was hit, how much damage it was, whether it was flak or fighter or what. So they would do these great maps on each aircraft, and they fed it into the first computer systems to develop this map of where the B-17s were hit most often. So they have hotspots. So the idea was, all right, let's have a meeting and let's talk about it. Let's up armor these regions that are hit most often. So everybody's like, okay, that's a great idea. Let's up armor these wingtips and the tail. And they're like, all right, everybody, we're doing this. Let's go forward. And somebody puts their hand up and says, no, it's a terrible idea. It's because what you're looking at is the planes are hit there most often, but those are the planes that make it home. Those are the parts that are least important. The planes that never come back, that don't come back where they're hit, is what's important. So they kind of did the reverse of where spots on these planes that are never hit. And they found out that, you were looking at this selection bias. Your native selection was stopping your ability to tell what's really driving this problem. So when we start trying to understand the microbiome, that's orders of magnitude more complicated than that simple problem. So what we try to use, our logical, teleological approach, doesn't work the way we think it should. So we're still trying to figure out who are the big players, who are the good players, who are the bad ones, and how much control does that animal genetics have on that microbiome before we can say, here's a good one. So there's not a handbook of what works and what doesn't. And I mean, a lot of companies will tell you, oh, our stuff works, nobody else's does. But there's really, to me at this point, I've got a handful of companies that I trust with what they do. And, you can kind of get a feel for those companies over time. I mean, how about you, Michael?
Yeah, I totally agree with everything you've been saying, Todd. We talk about a healthy microbiome. I don't think that we've defined that whatsoever. I think it's very naive to say that we know that in a calf. For example, is that the digesta? Is it the epimural microbes or the microbes that are attaching the digestive tract? I don't think we really know. And the way that we've been characterizing it is, still at phylum, genera, like levels. sometimes family, but not a lot of depth to a lot of the work that has been done over the last 10 to 20 years in this field. So I still think that that definition is still not truly defined. And I kind of got frustrated with it doing microbial work, and then you start profiling, and I am lucky enough to work with some excellent microbiologists that can help me with that. But I found more information on looking at the host. And, of course, you have to characterize the microbial profiling and do that to the best of your abilities with the samples that you have. However, I think we need to look more deeply at the host and the physiological mechanisms of the host and how they're responding to the probiotic or the microbial based solution of any sort, to truly understand when it's going to work and when it's not. And I think that's probably the next step of research, and that's what we're all trying to figure out now.
Yeah, I agree. And, about 10 years ago, I'd have said that's a stupid thought. But, that paper, and I've used it a lot, the Jai Freema paper from like 2017, where they look, and this is a great paper, they were looking at lactating cows. And instead of looking at the microbes, they were looking at the gene expression by that dairy cow. So they gave one group of them a probiotic, one group didn't get it, and they looked at gene expression in those cows over about a 60-day period. And they found there were 11 that you feeding a probiotic, and it was a live probiotic, it affected the regulation of 11,000 different cattle genes. So nothing about the microbes. They don't have any microbial real information there. But those 11,000 genes were in 87 different pathways. A lot of them related to growth hormone, inflammatory response, and systemic inflammation, which starts to make sense, because some of the work that you and I and Lance Baumgart have been playing with, looking at that widespread inflammation. Again, there's more smoke to it. We've been trying for years to get that answer, but we haven't been able to do it yet. But just seeing how those microbes directly affect the animal population and the animal genes and expression is amazing. So you've converted me in the past few years to looking more at that physiology effect of it rather than just the microbe effect.
Yeah, I think it might reflect the way that we actually characterize the microbiome in our research. But I was really surprised too when the first studies that we were doing were showing responses on the host when we were probing certain mechanisms, and not a lot of difference from a microbial standpoint in digesta or tissue attached microbes. But the actual host itself and their immune system and their physiology was changing, and actually a
Yeah.
little bit more reliable to characterize, at least with the
Well,
numbers that we're using in our research.
and I think that's going to come down to what are the end products of that fermentation more than who's there. So, I mean, your work has really convinced me, and I think you'll see it in our data and our papers in the next few years that we're starting to try to shift that way a little more, that we'll get that general microbiome. But I think we're trying to split the hair finer than what we can read the difference in, because we can't really measure strain differences, and those strains are important. So I think we need to look more at those biochemical endpoints and things like this hormone signaling. A lot of the quorum sensing molecules microbes use also work at the animal level, and that affects that physiology. And if you've got a lot of acid and low pH, that's going to give us leaky gut. So there's more room for those chemicals to cross that barrier and cause impact in the host physiology.
So one thing I think is getting towards one of the questions I have is, and we've just started as a company getting into this space as well. And part of the reason for at least myself and Dr. Sanderson, one of our colleagues at Balchem, has been really the nutrient absorption side of the equation, right? So we're, at Balchem, for example, we're a nutrient delivery company. To us, the next step is that, great, you've delivered the nutrients in a better package. How do you get those nutrients absorbed? Whether that's a ruminant or a monogastric, doesn't really matter. And that I think is where we need to really push that envelope and have a better understanding is how can we really unlock that biology of the animal through that microbial population and increase the absorption. So having said that, how how much further do you think we can go? I mean, if we really get this locked down, so we're at fairly low efficiency of use of nutrients from a ruminant perspective, right? Whatever number is 30-35%, I think sometimes. But could we get that to 50? Could we get it to 60 percent utilization if we really start dialing this in? I mean, I think there's so much of a benefit here that we're just scratching the surface on. So just curious your thoughts, and feel free. One of those thoughts could be you're completely off base, Ryan, and go do something else. So
Yeah, I'll speak from a calf standpoint. I don't think absorption has really been studied that much, and same within a cow. I think we've been really mainly focused on digestion and then the inflammatory response of the animal. There have been some studies in calves using yeast and lactobacillus showing changes in gut structure and the development of villi, even papillae in the rumen too. So I think that's one, you know, somewhat high level way of looking at how some of these microbial based solutions can actually improve overall absorption of these nutrients, at least from a surface area standpoint. But I think the nitty gritty of the molecular mechanisms, I think that we're really far away from truly understanding how it can affect absorption. I think digestion, we're getting pretty advanced in that, of course, with especially fiber digestion and different starch digestion. But I think from an absorption standpoint, I think that's somewhat lacking. But I think Todd knows a lot more than I do in this area.
Well, the absorption, no, you know more than I do on that. I'm a microbiologist and I pretend to be a nutritionist a lot of the time. Um, and a lot of that comes back to the stuff that Baumgart's doing and others like it. Lance does kind of the best work I'm aware of looking at that gut integrity. Because if your gut's not solid, you're not going to transport as many nutrients in that absorptive capacity. And he's pretty much heavily on to looking at that hindgut structure. And I think that really is more important than what I ever thought. He's kind of, again, he's brought me around to his way of thinking. But we probably shouldn't tell him that. That would give him a big head. But you know how Lance is on things. But it's one of those points that I think he's right and that I was wrong over the years. That I think that hindgut absorption is a big deal. And I think if we can make that absorptive capacity, both ruminal and hindgut, better, then we can absorb more of those nutrients. Because we're kind of at a point now where if we take a probiotic approach or an enzymatic approach and just break down this forage feed stuff very rapidly, we lack the capacity to take that much of the VFA up. So it's going to have to go hand in glove, at least I think it will, between the animal and that microbial population. And, we're trying to figure out if we can, a lot of our focus is trying to figure out if we can manipulate that microbial population to actually increase that degradability in the rumen rather than looking at digestibility, like what the microbes can do. And then is that affecting our animal performance? If not, what's limiting that absorption or that absorptive capacity? Is it pH? Is it too much starch digestion impacting your papillae length? Are we not getting those calves started early enough? I mean, in my class, I use a lot of your work, Mike, looking at calves and the papillae length and, how we want to get them started with a little bit of starch, a little bit of forage early, just getting those papillae to grow and getting them jump started, because then those calves can succeed later on really well. And especially with all the stuff with dairy on beef and all the problems that we're running into now with liver
Mm-hmm.
abscesses, that I think goes back to the way a lot of those calves are reared right off the bat.
Yeah, I totally agree with you. And actually, that's what we're working on right now is trying to untangle some of this with the huge problem in liver abscesses that we have in the beef on dairy field, not just Holsteins, but also crossbreds. It appears,
Yeah.
based on some of the first theses that are coming out and some of the first magazine articles, it's a severe issue. And I agree. I think it's coming from early life, and that's what we're trying to do. But you made an awesome point about we've been so focused on the rumen, but a lot of this stuff is probably happening in the lower gut, being the small and large bowel. And I would argue that it's as equally as important as the forestomach. And we never really pay close attention to it because it's hard to study in ruminants. You can sample the rumen and it's relatively consistent, but then you get to the small and then the large bowel, and every single meter is different. And what do you sample in the small intestine? Are you going to do 3 different segments, 4, 5? They're all going to be different with respect to host impact of your microbial treatment. So it's very challenging to do so. But I really like what you were talking about with leaky gut. It's something that, of course, we've all been inspired by Lance to study more. But I think that even that, we know, we just presented some data where, of course, like in early lactation, if you look at leaky gut using chromium EDTA as our typical marker, it will increase in early lactation compared to the dry period. And it remains still elevated even into early lactation. But is chromium EDTA or even like a marker for paracellular or the transport between cells really the best way of looking at this? And is it kind of trivial what we're looking at? Is transcellular just as important? So these absorptive mechanisms, I think we still have a long ways to go, but we know that there is a problem. Is it linked to systemic inflammation? I think it's a really good hypothesis, but I don't think that we've really shown that in the literature yet. But it's that so many people are studying this now because I think that there's a lot of opportunity.
There is, and Lance did some cool work, and I was kind of hanging on giving advice from the corner with his grad student Megan Avieda, and she did some really great work, had a great model for hindgut acidosis on the preliminary study. I mean, it looked fantastic, where the hindgut pH was down to like 5.1, and it was doing great. So then we do the exact same thing in cattle and bigger group of cows, pH of like 6. It just didn't work the second time. And
Mhm.
What's different? We have no idea, and it's just incredibly frustrating. And he's pumping all this glucose down into the hindgut. Sometimes it works, sometimes it doesn't cause hindgut acidosis. Sometimes it causes leakiness, sometimes it doesn't. What's going on?
Yeah, and is it really the pH? Is that the problem, or is it something else? I think we need to open up our mind, especially even with ruminal acidosis, is what I focused on for my PhD. And is it? I know some cows with acute ruminal acidosis that had zero health problems and were producing the most efficient animals in the study. I think that it's probably a little bit more than just pH, and we need to look at some of these other bacterial-based toxins. Of course, LPS comes up in everyone's mind, but yeah, I think we need to change our mind and the way that we look at a lot of these challenges in the dairy industry.
Well, and I think it's got to be some kind of signaling molecule with the lactic acid. It's going to be multifactorial
Hmm.
of that lactic acid at a certain pH and some other signaling molecule that basically then opens up I guess it's the space between the cells.
Yeah,
That tight junction would be my guess, but I don't know. You may be right. Maybe just that transcellular, dumping into the bloodstream.
yeah, and we don't really look at transcellular, so the absorption through cells, and I think that that's requires some merit. Uh, there's probably a lot more of that happening compared to paracellular, especially in the rumen where you've got like a stratified squamous epithelium similar to our skin. That's sometimes 20 layers of epithelial cells and many different layers of really tight junctions, especially in the granulosum. So yeah, I really question if it's really happening in the rumen. And it's kind of interesting looking at this liver abscess work too, that perhaps it's not just in the rumen like what we thought for the last 20 years, that hey, maybe it's happening in the small intestine as well, or even the large intestine. So yeah, I think we need to reevaluate everything. This sounds. I'm sorry for sounding like a professor who's like looking for funding. I'm sorry, but
Now, I'd love to get you together with Rand Broadway and Kristen Hales
mhm,
to talk about that, because that is something that they've been looking at with, along with TG Nagaraja at K State. But Broadway and Hales are doing some great work looking at those on the beef side of things,
yeah,
but using those little dairy calves, dairy on beef calves. And, I think they're figuring out a lot of the mode of action. And the interesting thing that fits into this is they're seeing a lot of salmonella in those abscesses,
hmm,
which starts to make sense given its ability to be a transcellular pathogen as well. And maybe it's damaging those cells enough to bring others with it across that ruminal epithelium or hindgut epithelium.
Yeah.
yeah. It kind of makes sense when you think about it. Why they would have more liver abscesses, the dairy or the crossbreds. They're not being weaned at 6 to 8 months. All of a sudden, we put them on a high starch diet from basically the first month of life, and they stay on that for their entire life. That's got to compromise gastrointestinal function. Such a contrast between a beef calf on a cow getting some fiber, a little bit of sugar from the grass, and maybe some creep feed, but predominantly milk as its nutrient source, at least for the first 4 months of life. So, yeah, lots to do there, and looking forward to sinking my teeth into that over the coming years.
Yeah, that's going to be some fun things to see what you come up with on that. I'm kind of excited, and part of me is like, man, I wish I could do some of that work, but we've got our strengths, and yeah, I would fail in those studies.
No, Todd, we got to talk after. You're going to be a collaborator, okay? I didn't
Okay,
say
cool.
that.
I'm good with that. Yeah, no, I'm good with that.
Yeah,
You know
real
that there's an
science
open door
exchange
here.
builds collaboration. So,
Yeah,
Without a doubt.
exactly.
We have seen that in the past as well.
And
Absolutely.
that's a great thing about this field is everybody wants each other to succeed. I mean,
yeah,
we throw these names around, but these are people that we can all call and go, hey, what the hell is this? And they'll answer, they'll voice their frustrations. And, in this world, all these scientists and nerds really want to help the producer succeed. And I mean, that's one of the things that marks us as very different than true microbiology. It's why I stay out of the micro field. They're looking for problems and they're trying to cut each other's throats, whereas we're a lot more team oriented.
it's funny that you said that because I said the same thing to my students at ADSA this year. Is that we are so lucky to be in this field as dairy and ruminant researchers in general because we're genuinely passionate. All of us are very genuinely passionate about making a difference on a farm for the farmer, and egos don't really play in to how we work together. It's incredible. We're really lucky to be in this field. Hmm.
I, I think what's exciting here is, I mean, there is so much. Sometimes I feel like in the dairy field and the ruminant nutrition in general, we're almost like ships passing in the night, where we have the true ruminant nutritionists focused on the nutrient side. We have the microbiologists, focused more on the micro side. You've got the veterinarians focused on the health. You've got the agronomy folks doing, well, we don't even know what they do, right? And I think we're finally getting to a point where we're all realizing that, hey, we're all in this together, and the more we talk and collaborate, then we can really start making some significant headway. Which leads me into my next question, which is, where do we bring the agronomists into this and the folks growing the crops? Because I think we always just say, hey, we're dealing with what we get, but now that we're looking at this tie-in between the biology, obviously the microbial populations, and could that be impacted by the feeds that we're getting handed to us? Where do we bring that into the equation and really start looking at that aspect? Because I mean, to my knowledge, I don't think we've really even talked about that. We just assume it's there and
We
we
Yeah,
feed
it's
it.
a great point. and we've been working on that. We've got a research lab, our research center down in Tifton, where Jennifer Tucker is, and Jennifer's doing a lot of work. She's a lot more grass oriented than I am, so she's been doing a lot of that work. We've got Nick Filipov here, who's done work on the fescue with endophyte or non-endophyte infected, where we were looking at what does that do to the microbial population and those end products and all that fun stuff. So there's people that are doing it. Brandon Smith at Auburn is working on that as well. So there's people doing that. The problem with it, and I always, you'll love this, Mike. I'm always like, well, why don't you guys don't do this? They're like, okay, it's going to take us a year to get the stand of grass grow. Then it's going to take us 3 years, assuming the weather is perfect all 3 years, to get the first data. And then, it's like 5 years per study. And once they start saying that, I'm like, oh God, no, we can't. No, we can't even try this. So I don't know how Jennifer and Brandon and those guys get this stuff done. It is just such a nightmare to do a study like that where it takes forever to do things. And, you and I, Mike, we're like, yeah, 6 months, that's a long study. God, it's so terrible. So tired. And these guys are doing 3, 4, 5 year studies.
Yeah. We have to
I don't
stop
know how
hanging
they
out
do
with chicken researchers and swine researchers.
it.
They make us very jealous as ruminants, but then we have to look at the crop side. It's totally true.
Even slower,
I,
yeah.
Yeah.
I think there's all kinds of potential there too that hasn't been tapped. And we
Mhm.
have been in our own silos, and it's really great to see a lot of people, even animal scientists, reading a little bit more about soil microbiology. That's how I really got into very interested in microbiology, is looking at the soil in undergrad and realizing,
Yeah.
wow, this is completely active, and we have no idea really what the impact is. And but I
Yeah. I
think
saw
yeah,
something
you go
on
ahead.
soil health like that, and it was just, it's amazing how complex that is.
Mhm.
Well, yeah, and we're talking about calves and setting their microbial health for the rest of their life. But the same thing could be happening in the soil, right? If we're setting a good soil health, that's helping everything else that's being grown from that soil. And I've often wondered, over time, we've, think whatever in a native cow, so to speak, she's out consuming grass, she's eating all kinds of stuff. She's just not eating a pure stand. But over time, what have we done? We've created pure stands of whatever we're trying to harvest, and maybe we're actually shooting ourselves in the foot, so to speak, because we're killing off some things. Could be soil microbes, but it could also be what we would term weeds, that we're actually having a beneficial impact to the nutrition standpoint.
Yeah. Empty a cow's rumen in New Zealand, Australia, you'll quickly figure out that the soil microbiome is there because
Mm-hmm.
the ventral sac is completely full of soil. Uh, but I think the clearest
Didn't know
where,
that.
yeah, I've emptied cattle in like rumen fistulated cattle in New Zealand, and the bottom of the ventral sac is all soil, just heavy sand. Um, Yeah, it's remarkable actually. Papillae are
Huh.
still there. It doesn't really affect them, but yeah, that's what happens. So I'm sure we're missing all kinds of things on the soil, but the plant side, I think where we've kind of shown that it's work out of Canada and Alberta by Tim McAllister is how like an inoculant for forages can impact, of course, the amount of bacteria that is ultimately fed to an animal, and that could potentially impact its physiology as well. So I think that that's closest where we are to the industry is looking at inoculants and how that can affect the forage. Lots of opportunity there. It always kind of boggles my mind when I think about how many microbes are in silages, and we just kind of add a little bit in a dietary treatment. We kind of forget about all the inherent microbes that are in forages, and so again, more work to do. And that's probably why we're seeing so much variability as well, or one contributing factor to some of the responses and worldwide.
And maybe if we step back and consider the soil health, maybe we're going to inoculate those forages with some microbes that could help them during the fermentation process. When you're ensiling, maybe you end up with a better product if you're starting with some microbes that might help facilitate that process just from when you're growing it.
Mm-hmm. Definitely lots of opportunity to tear down those silos between all the fields.
Full pun intended.
Mm-hmm.
Yeah. Maybe kind of switching gears a little bit, Todd. During your presentation, you talked about different animals that have a different microbiome, and I don't know if you meant just rumen or lower gut, but the fact that the least efficient cows tended to have a more diverse microbiome, as opposed to your more productive cows had a less diverse microbiome. And I found that very curious and interesting. Would you mind kind of expounding on that just a bit?
Sure. Yeah, that was a study that they did at the University of Illinois back in 2016. And, the dogma has always been that more diversity is necessarily a better microbial population, which makes sense when you think about it. More diversity is good. Sure, I can get behind that. But what they found was the most efficient milk producing cows, when they looked at that microbial population, especially in the rumen, that lower diversity meant a lower diversity of microbial end products, of metabolites, like VFAs and such. So compared to the more diverse population where you had more end products that weren't necessarily used by the animal. So the analogy that I use is it's building a car. So if I'm building a car in my garage, it's going to take me 5 years to build one. But if you train me to put 2 screws on 2 different bolts, we can sit there on an assembly line and a group of us can make a car in a couple of hours. So it's that difference in efficiency by being focused on what that job is. And it goes back to the point that Mike made earlier about looking at those output. What are those metabolites? What do they look like as far as a profile of what the animal can use? So it's about being a little more focused in a microbial population rather than being completely broad and doing a little bit of everything. So, when we talk about this microbial population, the ruminant evolved to be able to adapt to different feed systems where they can eat a little bit of this and a little bit of that. So that diversity, that broad diversity, was always a benefit to the animal for survival strategy. But what we're feeding them now for production is not a varied diet. It's a very similar diet. There's a lot of homogeny to that diet day after day after day and across farms. So we are providing this artificial selection for a more narrowly or lower diversity population. And what is that going to have on our animals long term? Number one, because we've got millions of years of evolution for them to be adaptable. And adaptability and efficiency tend to be negatively correlated. So we're taking these highly adaptable animals and trying to make them efficient. So we're fighting a tide in a lot of ways.
Interesting. You mentioned metabolites, and during your presentation, you also talked about malate and fumate. Found that very interesting. Could maybe expound on that just a bit? It seems like there might be a commercial opportunity there, and I don't know if anybody's taken advantage of that or not.
Oh, that one is something that goes back to, we're talking about the old stories. Malate and fumarate, I worked with those on my master's degree with Scott Martin before he left working in this field. And malate and fumarate are dicarboxylic acids that fit into the TCA cycle. So plants have them in a lot of when they're growing rapidly. And a lot of their, those, when we talk about the sugars, those are just simple compounds in that TCA cycle. But bacteria like Megasphaera elsdenii and Selenomonas ruminantium that are lactate utilizers. So they'll take lactate and convert it to propionate. Now they're the guy that comes in to a party, sees a bunch of donuts, licks the donuts so no one else will eat them and they can come eat them later. So that's their strategy, is then they're taking lactate to propionate. Well, if you add malate and fumarate, because you're adding these intermediate metabolites in the pathway, it causes that pathway to spin faster. So it increases the lactate utilization rate by those lactate utilizing bacteria. So it speeds up their metabolism. So you're shifting lactate to propionate faster. And it's a really cool compound to work with. Malate was the one that was the most efficient. And it's like, we bought a bunch of it from Japan years ago in 50-pound bags that was the end product of aspartate production for amino acid companies. And it was a waste product they had. And I think they still do that we can use. The problem was it's a little bit hard on machinery because it's an acid and it'll heat up augers. But companies started looking at it and it just never went anywhere as far as being additive. Because in that era, again, this is 30 years ago now, that era, people didn't really think about that approach. Well, we also discovered over the years that a lot of our postbiotics contain malate and fumarate naturally. So they stimulate that lactate utilizing population to go ahead and grow faster and utilize lactate more rapidly. So it causes a shift in that microbial population simply by giving those guys a competitive advantage. So I recommend a lot of companies take a look at what their composition of their product is as far as containing malate and fumarate. And, you can use that as a hook to understand what's going on in that population. So it's really something very cool. And that's where, we start talking about eubiotics as encompassing organic acids. This is where things like malate and fumarate would fall.
Interesting. Well, gentlemen, this has been a very interesting conversation. I have a feeling it could go on for quite a bit longer, but we're kind of at the top of the hour here, and I think it's time to call it quits, or call last call. That's what we say here. And with that, what I'd like to do is if each of you could just kind of give me, one or key takeaways, maybe for a nutritionist, maybe for the research community, that you think we ought to take away from this conversation. And Ryan, if you don't mind, I'd like to start with you. The economics of feeding Reassure precision release choline. Reassure is fed during the transition period, and because it's fed for such a short period of time, it costs just $15 per cow, and yet the benefits will continue to generate income throughout the year. Cows fed Reassure produce 5 pounds more colostrum, which pays for your Reassure investment on the very first day of lactation. Cows fed Reassure also produce 5 pounds more milk per day, every day. That means after the first day, every day is payday. Invest in Reassure during the transition period and recoup your investment on the very first day of lactation. After that, you got it, payday.
Sure, yeah. I think the takeaway is, as a nutritionist who used to be in the field, I think the takeaway is, for all you nutritionists out there, don't forget to have the conversation with your partners in crime, i.e. the dairy producer, whoever beef producer, whoever that end user is, the veterinarian, the crop team, and the management team on the farm. And make sure you're all working in coordination, talking, because that's really where the synergy is going to be, I think, now and particularly in the future, as we learn more about how this all coordinated biology is working together. And that coordinated biology is not just within the animal, but it's outside the animal. It's all those other factors that are coming into play, like we talked about, from the soil to the crops to the environment that the animals are in.
Yeah, thank you for that, Ryan. Mike, you've been a great guest. I'm glad that Todd asked you to join us tonight. Really appreciate your comments and insight. Do you have any final thoughts for the audience?
Yeah, for nutritionists and farmers out there, only use probiotics or any other microbial-based solutions that have minimum 3 publications showing efficacy in a high-ranking journal, not just any journal, but a Journal of Dairy Science or even higher. I think that's one of the main problems why we've received this, negative reputation in our marketplace, because we are actually feeding a lot of microbial based solutions that don't have enough research behind them. And I think it's critical. I think there's a lot of really great products out there. There's a lot of great companies. So I think I would pay attention to that and, measure it on farm. Make sure that you choose your metric properly. If it's a healthy herd, perhaps that's not the best way of looking at it. Maybe just focus your analysis on farm with the cattle that are experiencing some stress or some metabolic or infectious issues, to truly evaluate the return on investment on farm. So I think it's not foo-foo dust. I think there's great solutions out there, but you just have to make sure that it's a proven solution that from a company that's research-based. That's really critical.
Yeah. Excellent advice. Excellent advice. And Todd, I want to thank you for joining us. This is your first time at the pub, but I can guarantee it will not be your last. We'll plan to have you back here. You've been a great lecturer and a great guest here at the Real Science Exchange. Any final thoughts from you?
Basically all in mind. Be cynical about these companies that are coming to sell you things, because you ask the question, how does your product work? If they're like, well, it just does, be a little cynical. I mean, I know that sounds harsh, but a lot of the companies that are selling the foo-foo dust, they don't know how it works, and they don't really care. It doesn't matter. But the companies, like Balchem, that have put time in and doing science, they're going to have an answer. It may not be right because we don't know the answers, but they've at least looked into it to know what's going on and try to understand that. So that goes to Mike's point of looking at these research, and companies that say, well, our product works in these situations, but not these. Those are the companies I'm going to trust, because they're telling you when their product doesn't work. So they're giving you their real data. They're not just giving you cherry picking the good examples. So those are the companies that I really tend to trust and follow. And, in his point of what is your metric that you're using? What does success look like for you as a farmer? That's the thing to keep in mind is, what is your metric? It's got to be measurable. It's got to change. We all want to think about its profitability, and ultimately that determines if you stay in business. But profitability is a product of so many other things that you've got to focus on something bite size that this product can change, that's going to impact that bottom line.
Yeah, well said. Gentlemen, I appreciate your insights, appreciate your collaboration. This has been a great podcast. Looking forward to doing it one again as a follow-up. So I really appreciate you guys joining us here tonight. And to our loyal listeners, I appreciate you joining us as well, as always. I hope you learned something. We hope you had some fun. We hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests. So please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.